clearly not Cindy Lawley. She's far prettier than I am. Just before Cindy gets on stage, I thought I'd introduce myself. My name is Arjuna Kumarasuriya. I'm Illumina's District Sales Manager based in our Singapore office and responsible for giving our users access to our technology in the South Asia region, which is essentially Southeast Asia and the Indian subcontinent. It's an area, of course, where rice is a very important crop, and as a result, Illumina is very proud to be here at this conference. So this mini session, we're going to keep it fairly casual. So I'd invite you, if you're at the back of the room, to please come to the front. Cindy loves seeing your faces and knowing whether you like what she's saying or you don't, as do we. If you have questions, please pull them up. The idea will be we will take maybe about an hour for this session, or about half an hour for this session. But what we really want to do, and one of the main reasons for conducting this session, is to hear feedback from you. As Cindy will describe, Illumina is a unique company in that we have advanced genomic solutions in both microarray and next-generation sequencing. And as a result, we are uniquely positioned to offer a very synergistic set of solutions to address growing needs in genetic communities such as the rice genomics community. As she'll also describe, rice is a pristine genome, so it offers some interesting opportunities. It's a small genome, and as a result, the MiSeq platform, which Illumina is obviously finding is being widely adopted, is very well suited to the size genome that we're talking about here. So those of you who saw the leaflet, and the last thing I'll do before I step down and let Cindy take the stage, there was a flyer saying that you have the opportunity to win a MiSeq run by attending this workshop. All you have to do, my colleagues from Bioactive, our representatives here in Thailand, are busy walking around handing you a couple of sheets of paper. One of those sheets of paper is an entry form. There's some questions there. We'd just like to know about your interests. We're obviously very keen to find out how we as a company can better address the needs of you as rice researchers, particularly in genomics, moving forward. And that's speaking to solutions both in arrays and sequencing. So that questionnaire lets us understand a bit more about your needs. There's also a couple of questions at the start. Cindy, during her talk, will be revealing those answers, but if you stick around, I might give them to you for free at the end of her talk. The idea is that at the end of the session, please find myself or the lovely ladies from Bioactive or Cindy even. Give that form back, we'll collect that, and hopefully we'll announce the winner before the end of the conference, who will essentially get a free run. And this will constitute a whole run on a MiSeq. We'll help you with the sample preparation. The sample itself, or the library itself, will be sent to our facility in Singapore. We'll sequence it, assist you with data analysis, and provide you with what's provided. And hopefully that will give you an idea of the power of the platform, and also give you some data that might be useful in perhaps acquiring your own. Other than that, there's no other housekeeping notes to mention. As I said before, please do put your hand up and let us know if you have any questions. And if you do have any questions from Illumina and you don't get a chance to catch us, pardon me, at the bottom of this entry form is a small space for you to address any questions. And what we'll do is ensure that an Illumina representative from whichever country you're from will get in touch with you and address those questions you have. Okay, so that's it from me. Thanks very much for your time, and thank you. It's great to have such an audience at the end of the day. But without further ado, I'll ask Cindy Lawley to come on stage and give us our presentation. Thank you, Cindy.
So I want to thank you very much for that introduction. That was absolutely beautiful. I don't get to reveal very often where I did my PhD, but I was pretty proud of, I worked on fish. So I was at the Scripps Institution of Oceanography before I joined Illumina. So thank you very much for that. And as Arjuna mentioned, you should have received by now a form to fill out. And in that form, we'd love to know what your plans would be if you're interested in winning a MiSeq run. So it would be greatly helpful to us to facilitate your research. So my role is as a consortium manager But today I'm going to talk about not just Consortia, I will touch on Consortia, but I'll talk about sequencing as well as arrays, and Consortia or private projects can leverage both of these technologies, or all of our Illumina's technologies. A little bit about me. After I finished my PhD, I started at Illumina, and I've actually been at Illumina for about 9 years, going on 9 years. It'll be 9 years in 2013. And so background on the positions I've held at Illumina, because I've changed roles through that time. When I first started at Illumina, we were a genotyping company, so we didn't sell sequencing then, and you probably hadn't heard about us. But for the first 4 years at Illumina, I did genotyping on arrays for customers, and so I was an internal scientist. And so on a daily basis, I would look at cluster files like this, which represent genotypes. So the AA cluster, the AB cluster, and the BB cluster. And actually, in reality, my job looked more like this, which is not quite as pretty a genotyping example. So it was difficult sometimes, especially when you have ascertainment bias, like in maize, for example. Here's another example of one of those difficult clusters to interpret. And then my next 2 years at Illumina, I worked in the sequencing side of the business. So by then we had purchased or acquired Solexa technology. There was a merge between those two companies, and I moved up to the Bay Area in San Francisco to work with the Solexa technology. And so we delivered data to customers, and we looked at a lot of adjuvant traces on libraries that went into sequencing. But most of them actually looked more like this, which is a degraded RNA library, or like this, which is a library prepped for sequencing that has a bit of a large distribution in size, which makes it challenging to go onto the sequencer. So that's some of my background. And for the last 3 years, I've worked supporting the sales team where opportunities arise where we can facilitate multiple groups using the same tool, so these consortia. And it's interesting to see since 2009, when I moved into this role, how many species communities rely upon working together in order to get more research done and to communicate using a common language. So that's been very exciting to see. Here's an outline of my talk. So, as Arjuna mentioned, I think Illumina is uniquely positioned because we can help you get a holistic look at the genome, the rice genome, or whatever other genome you might be interested in, including the human genome. We've got both array technology and sequencing technology, and I think they can be very complementary. They can also be competing, and so the message really is, we've got to figure out what's the right price point for your project, and what are your project goals, and what's the quickest and easiest way to attain them and get funding for them, right? I'll talk about harnessing the power of arrays using the Ag Consortia process. So this is the role that I've been in for the last 3 years. And we'll give a little bit of a sequencing technology update. So this is where the answers to those first 2 questions in the MySeq survey will be. These are about the throughput. And then we'll talk about what's next for rice. And the what's next for rice is really about you. It's about what you think is next for rice, or what questions you have for us about how we can facilitate the moving together that I'm seeing in all of these consortium-driven projects that the rice community is involved in. It's really pretty exciting to see some of these collaborations that are going on. So as we mentioned about Illumina, we do have arrays and we do have sequencing. And so I just threw together some questions that help people decide whether sequencing is the right technology or whether genotyping is the right technology for any particular question. And so just for your consideration, I put them up here. So some of the questions you want to ask is whether you're hypothesis driven, or so do you have a priori information about what you want to query? Rice is a pristine genome, so you may have loss of function SNPs or must have content, which is the second criterion here. In that case, an array approach might be a favorable approach. How pristine is your genome? Rice is a pretty beautiful genome. At least the first genome published has been beautiful, and I know that there's goals to have additional assemblies made for other lines in rice. I think it's Nippambari is what comes to mind. Isn't that the published reference, right? So as you get further away from Nippambari, you're going to have challenges with ascertainment bias where an array approach may not be as favorable. So those are the kinds of questions you want to ask yourself when you're evaluating the approach to take. And Illumina can help with this. We've got a lot of experience with sequencing and arrays, and using one or the other or a complementary approach, sequencing fueling array content, that can be very helpful, I think, in deciding what you want to do. We're not going to sell you something that isn't going to make you happy or get you to where you need to be. And this is actually a good time for me to mention that Illumina has, Rice Genome is special because it's pristine. Illumina is special because we have an ag program. So we actually have devoted people that are specialists in agriculture. We have a whole team that works on that. And Arjuna works very closely with our team. He's not only ag, he works in the human space as well, so he can transfer information from what they're doing in the human genome to agriculture. But Illumina has a lot of specialists and information about agriculture species, so I think that we can partner with you. Other questions, how many samples do you have? In many cases, the more samples you have, an array approach can be favorable, and it all comes down to price in many cases with agriculture, which I see over and over again, and I'll talk about that more in a minute. Price per sample that's tolerable, what do you need to hit in price per sample to get your research done, to have the power that you need to detect your genome-wide association, to identify your haplotype blocks and make your trait associations that are essentially the holy grail of molecular-assisted breeding. What are your informatics capabilities? In many cases, if you don't have a solution for informatics, sequencing might be less favorable. Can you tolerate ambiguity? In this question, I mean, with sequencing, sometimes you have to, depending upon the depth you can afford, you have to tolerate not knowing if you have a heterozygote, if there is a heterozygote present. And in that case, in the case where you're just looking for fixed alleles for your two parents, that's okay. You don't need to know about heterozygotes. And so sequencing might be an advantage there. So it's really just not a one-size-fits-all, not one is better than the other. It's a what's right for what you need to accomplish. Although I will say, right now funding agencies think sequencing is super sexy, so it gets a lot of attention, and that's something to consider as well. Interested in whole genome versus focused regions. Is there epigenetic aspects to your question, in which case sequencing might be favorable? And are you in a discovery mode or a screening mode? Now, what I mean by this is Illumina has seen that there are applications for agriculture where you need to discover what you need to know about the genome, and then you need to implement it in a breeding program. That transition from discovery to implementation or deployment is a really key return on investment requirement to demonstrate value. And so we talk about discovery, develop, deploy, and those are steps that you'll need to do if you're trying to assist in breeding. If you're academic, then maybe discovery is where you stay. But often you'll need to translate that to those who are talking to breeders. And so we want to understand that better and make sure that we can support you in being able to do that. Because if you can demonstrate return on investment, then you can make use of the next generation of sequencing technology, the next generation of molecular assisted breeding. And that's what we want. Okay, so that brings me to the logo, Discover, Develop, Deploy. And this is one of those that you're going to see at the Plant and Animal Genome Conference, both in San Diego as well as PAG Asia, which will be in Singapore next year. So we'll have presence at both those meetings, and we hope to see you there. And this is a slide that just shows you examples of the discovery phase. Many projects have gone through this discovery phase, especially since the commercialization of the next generation sequencing technology, not just Solexa technology or Illumina technology, but 454 technology. All of these methods of sequencing that have moved beyond Sanger sequencing have made possible SNP discovery projects never thought possible before. We were working with microsatellites in all these genomes before, which can be very expensive and hard to scale. So some examples. The bovine project, this is the project that I worked on with the bovine community to develop a SNP chip that was a commercial array of ours. And I'm sorry to put an animal up in a rice meeting, but it's a good example of a method of SNP discovery and then deployment onto an array. We've also done a SNP discovery project in porcine, and that ended up being a commercial product as well. We got 2 publications out of both of these. We did a publication on the discovery of the SNPs, and then we did a publication on the development of the array. So it was quite rewarding. And we got USDA awards for each one of these projects, which have revolutionized the breeding programs in these animals. Rainbow trout, another example, and these are not comprehensive. There are examples I'm not showing here. Soybean, very informative. Grape, we now have a grape array I'll talk about in a minute. Maize, and while the maize project, the SNP discovery and selection was going on in maize, Ed Buckler was working on the genotyping by sequencing Cornell method that is now so popular today, the reduced representation method of indexing that gets the price per sample really low for genotyping by sequencing. Pine, another species. Pea, wheat, we've got several panels of wheat SNPs. So polyploids are accessible. We can do polyploids. Forest tree genomics, eucalyptus is included in that, and chicken, eggplant, peach. Peach is a beautiful genome. That is a nice genome as well. Not as nice as rice, but a great one to work with. Apple, and not last but not least, there are also some OPAs, there's SNP discovery projects that we've heard about that have occurred recently, but there have been historical SNP discovery projects that have helped us with particular lines in rice. And there are low-density panels that Mike Thompson and Susan McCooch and collaborators have made available to the public and put out there to help with selection. And Erie uses those panels. So this leads to all of that revolution or knowledge that we learned during that phase of 2009 to 2011 and today has shown us that we can discover the SNPs, but often these communities can't afford the arrays that would be required to genotype routinely and do marker-assisted selection. And so what we've done is develop what's called the Consortia process. So the Ag Consortia process is a process that Illumina has created in order to help with logistics around developing arrays that will meet the needs of multiple individuals. So this way we can leverage the economy of scale. So if it costs a certain amount to make the array, if you can spread that cost over more samples, we can get the price per sample down lower and more people can use it. Because in agriculture, we see again and again and again that if we get the price lower, people will do more samples. They don't just stop with they use up their budget because they need to get that information and detect that the genome-wide association or the linkage or the trait association. So my job as a consortium manager is essentially as a logistics coordinator and a protector of confidentiality in those circumstances. And then as a result of the consortium process, Illumina developed many commercially available arrays. What this means is our marketing group actually paid to have the validation work done, paid to do the development project, and invested in commercializing these products and making them available in our catalog. But these are pretty big markets. Maize is a big market. There's lots of samples, lots of breeders. There's a lot of tons of maize, a lot of it, a lot of bovine dairy, a lot of use in dairy cattle. So they're big markets, and so we could make these big commercial arrays. But what we found were that we could really benefit communities that were smaller by doing the same thing, but just having lower sample numbers. And so if we could keep our costs down, we could make the price accessible to those communities as well. So since basically 2009, we've developed over 25 different arrays for different species. I'm not sure if you can see the species, but potato, tomato, apple, pear, peach, cherry, maize, wheat, 2 wheat. One is a 9K, the other was a 90K, 90K. Barley, ovine, a sheep, chicken, equine, horse, oat, potato, grape, cat, feline, dog, couple of bovine arrays, brassica array, that's a beautiful one. We should hear a lot about at PAG this year. Goat, yeah, a lot of really exciting products we've worked with the communities to develop. And so I guess my message here would be that we're pretty experienced at working with communities, at protecting confidentiality, and at making sure that the SNP selection best practices get transferred to new communities who want to do the same thing, because we want them to be successful as much as the researchers who are leading the projects do. So when we work with communities to do these projects, we also do our best to make sure that they stay in our catalog, that we have enough bead pool to sustain them over multiple years, which is also something that agriculture customers need, that people in the human research community don't often need. So those products don't tend to stick around for long, but we've had these in our catalog, or in our portfolio for a couple of years now, some of them. Also, the number of SNPs vary for these because the number of SNPs needed was perceived to be different based upon the genome or haplotype block structure, or just what could be afforded. And so the nice thing about the fact that we've got sequencing as well as array technology is that we have something called add-on content, and groups can go back to these bead pools and add more SNPs for a very small price. And so the flexibility of being able to take your 8,000 SNPs in potato, do another bead pool and add 2,000 more SNPs is really a great flexibility. Or companies might want to come along and put their proprietary content on top of the base content that is shared by the community, in which case, say a seed company in Europe wants to do a study on potato, they may have 2,000 SNPs that no one else is allowed to see. We make bead pool in liquid format. We take the base content of the potato chip that is public, and we put them together in a combined bead pool that's just for that European seed company. So this is called add-on content, and it's worth exploring, because with sequencing and add-on content, there are endless possibilities for the density and the depth that you can do with genotyping technology, if that's the right fit for your project. So that's the add-on content. And then we've got some projects that are now in progress. So we always have consortia that are in progress. So we're working on a cotton chip that's expected to be available in 2013. So if you're interested, please talk to Arjuna or me about that, or your colleagues at Bioactive. Melon is in progress. Pepper, we've got a second generation of apple chip that has higher density. We're doing a high density sheep chip and a soy chip. And then because the thought leaders in each one of these communities are helping us with informing what the right content is for the community to get the best turnout, so that the most samples can be used to spread the price over, we try to protect those thought leaders from all the questions that might come up around how we develop the projects. So what I do is I work with them to create FAQ documents, or frequently asked questions documents, and we distribute these documents to new customers who might be interested in these arrays. So here are some examples. There's a tomato one, a potato one. These are available on Illumina's website, or you can call tech support and ask about any of these publicly available SNP chips. Apple, peach, and cherry, grape, and these answer questions about how the SNPs were discovered, how the SNPs were selected, how we pared down the content to make it most relevant for the most number of people and achieve the most research goals. Brassica. And then there's an FAQ document if you're interested in being a thought leader. So we're certainly interested in helping develop maybe a low-density rice chip or even a mid-density rice chip, something that's not necessarily covered or available now. If people are interested in that, we can give you this FAQ document. We can sit down with you and talk about what you think is needed, or we can steer you to genotyping by sequencing if that's going to be the cheaper solution based upon the number of samples available. And the reason the consortium process works, as I mentioned before, is the economy of scale. So essentially, when we make a bead pool for just 1,000 samples, the price is going to be higher in every case than if we make a bead pool for 10,000 samples. I don't know if I've got a laser pointer, do I? I do. So you can see for 1,000 samples, the price per sample is higher than for 10,000 samples. This is a lower price per sample. And you can add more SNPs when you've got 10,000 samples to deal without the price increasing too much. So that's just a function of how our manufacturing process works and how we can spread that over more samples. So that's why the consortium process is so successful. And then my job in the consortium process is essentially to determine the market, how many samples are possible in that community, and manage the logistics and protect confidentiality of all the partners who are involved. And different densities of SNPs work for different communities, as you saw in the list of products that we have. So, for example, for the canine SNP chip, we made 170,000 SNP chip because their goal was to do crossbreed mapping. And to do crossbreed mapping, you need a pretty high density of SNPs. And it was super successful. We had a publication within the first month of delivering data on that panel. The Apple SNP chip, the first generation chip, they thought that 10,000 SNPs, basically 8,700 SNPs, was going to be enough for that genome, and they've discovered that they want to have higher density for additional research purposes. This is still used for screening because it's cheaper, but the 20K is been developed now for research purposes. And then, as we talked about earlier, the IRI rice panels are as low as 96 SNPs or 384 SNPs. So I think the 96 SNP panel is for QC, and the 384 SNPs are for particular lines. Actually, I think I have a slide on that. So this is actually the publication. Mike Thompson was the lead author on this, and Susan McCouch was the PI. So this, and I'm sure Mike would be better positioned to talk about this, but essentially they made these SNPs publicly available. They picked the panels for specific line crosses and particular testing of particular lines, and the reason that the SNPs were of that density was really to hit the price per sample that was needed to be able to screen the lines that they had in high numbers. And so that's why they went with the low density panel and get the biggest bang for their buck so that they could move through their lines pretty efficiently and make those available for others. So IRI has, as you know, a huge collection, and a subset of those are screened on these panels, and their crosses are screened as well. So here's their table in this publication and the various oligo pools that they've created and their purposes. So there's an indica, japonica, japonica, and then one with the wild type, and I'm afraid to pronounce that, rufipagon. I probably messed that up. Anyway, so you are probably familiar with the publication. And these are the outlined purposes for that design: diversity analysis, QTL mapping, marker-assisted backcrossing, developing specialized genetic stocks. But I recently have read this paper, which Susan was a lead author on, and I think she's leading an effort around making decisions about how to screen gene banks. And so out of this publication, I'm wondering if there might be opportunities for using these or a low-density rice SNP panel for documenting genetic identity, for identifying duplicates in the germplasm in gene banks, for tracking the lots, rationalizing active collections, for diversity that you might want to put into the commercial lines. Anyway, so this is just some of the background that I know about the SNP chips that are being used on Illumina's technology. And then I wanted to talk about price, because as we talked about with the oligo pools from IRI, it's really important that the price per sample is the right price to be able to get the problem solved, to get the question answered. And so this shows, and I'm not sure if you can see it, but it shows this is for potato, and basically I did a market survey of the people that were interested in using the potato SNP chip. I like to say potato chip. And $85 a sample, they claimed they would do 2,000 samples. $75 a sample, if we could get the price down to $75 a sample, they claimed they would do 2,500. But if we could get the price down to $65 a sample, they would do over 3,500. And so this just shows the fact that these markets are driven by price. In the wheat community, we saw that when we asked them about $117 a sample, we got very few commitments. But when we were able to justify with our internal team to allow for a price of around $60 a sample, we saw over 40,000 samples. So it's a huge difference in the way that these chips are being used if we can get the price per sample down. And we've seen array prices dropping. And so please just ask us and tell us what you need so that we can work for you on an internal justification to get our senior management to get at the right price point for the community. So this is really a call to help us understand what you need, because different communities are different. And we can make that justification to actually cut down on streamlining our internal costs and making sure that we can hit the right price per sample as long as we can see the volume from it. That's how we get senior staff on board. They like volume. And this is just more evidence that price per sample is really important for agriculture. So these were some of the fill-in answers in the market survey I did for potato, where they were saying they would love to test thousands of samples per year, but limiter is cost, or they've budgeted approximately a certain amount of money and they need the price to be below $50 a sample. And I keep hearing this over and over again, $50 a sample, $60 a sample, that this is a price point that's really important to hit for screening. And what I think is really interesting is that some of the data I've seen presented by the Cornell trained genotyping by sequencing groups is that if you can hit around $60 a sample, which is where in genotyping by sequencing, that's about sequencing at about 4x, that you're detecting almost 90% of the SNPs that are present at about that coverage. So that's about the price per sample where you're pretty confident that you've got all the information that you need. So that's the place where if we can manage to get arrays down to that price and still have them at the density that's needed for the community, then the analysis of the array data can be more simple, and we can make it accessible to those people who don't have informatics pipelines. So this is a place where both technologies can solve the goal, but one technology may not be right for a particular customer simply because of their capabilities. So this is where we can look at a community, talk to people in the community, and try and propose a solution that'll work for everyone. And then remember that add-on content is always available for people who want to have a higher density or have a particularly more rigorous research goal that requires more coverage, CNV, breakpoint detection, something like that. And this leads me to just touch on sequence-based genotyping. I'm calling it sequence-based genotyping, but it includes the genotyping by sequencing method by Cornell, also the RAD-seq method. So there are lots of publications on GBS and sequence-based genotyping now, or what we can call next-generation genotyping, right, using the sequencing technology and leveraging the low price of sequencing now. So there's skim sequencing. That's become very popular, where essentially you're just skimming at a low depth of coverage and doing comparative analyses, or just detecting alternate alleles fixed in your parental lines. PCR and hybridization-based methods include long-range PCR, solid or liquid phase hybridization, so Agilent's technology, for example. And there's restriction enzyme methods, which include the Cornell method, several of the methods of SNP discovery that I outlined earlier, and RAD-seq, which is popularized by Keygene and Florogenics. So here's some of the examples of publications. And Illumina, we're pretty familiar with the groups that have published on these, and so we can talk to the protocols and help enable you to implement some of these methods if you think they're right for you. So these are, anyway, I just wanted to, these are in order of their publication date. And so that brings me to talking about our sequencing technology and our current capabilities in sequencing and how they're improving. So around 2009, when I worked in the sequencing services group, this is what you needed to do sequencing. And we did a lot of business through the services group because we hadn't sold that many sequencers back then. And so a lot of people wanted to try out the technology and see how it worked. And so these are all the components that were needed, and it was closer to when it was developed than it is now. Now we've got it in pretty good shape. So now this is all you need. So essentially either a MiSeq or a HiSeq or a HiScan and sequencing module, and some data storage. And on the MiSeq, you've got a single instrument workflow where you have the cluster station is on board. And so this is essentially our sequencing portfolio now. And they're both so much easier to work with. And I will say that those GA libraries that people ran in our services lab, we shipped those out to customers. Those can still be run on any of our instruments today. So they can still be run on a GA2, they can still be run on a HiSeq. You can get more data on the libraries we made back then. We just have easier ways of making the libraries these days, but it's nice that they're backward compatible. So HiSeq 2500, this is the high output mode. So the beauty of the HiSeq 2500 is you can do it with a long run of 11 days, or you can do it with a short run of 40 hours, or even less than a day. Now the amazing thing about this is with the HiSeq, you could do 70 rice genomes in the high output at 20x coverage, and you could have those data in 11 days. It's a stunning amount of data and improving every day. On the rapid run, within less than 2 days, you can, within 40 hours, you can have 20 rice genomes done at 20x. So it's a big difference from the Human Genome Project, a 3 gigabase genome, which took 10 years and $10 million to run, right? It's stunning to me. I know you've heard all of this before, but it's still amazing to me. And on the MiSeq, you can do, well, let me go through and give you your answers on the questions, I think. So in 2011, when this was launched, when the MiSeq was announced it was actually during a PAG conference, or the AGBT conference, which was, I think, or ASHG, that was around that time. Our output was around 1.5 gigabases. Now our output is 8 gigabases, and these are the paired end 250 base pairs. So that's effectively 500 base pairs, plus you've got an insert, so your effective read length includes that insert, because you know the insert size, so you can add 200 to up to 600 bases in between there. So you have this virtual read length that's huge. So it really helps with doing assemblies, and at PAG we're going to have some groups that have done assemblies on complex genomes like Lawley Pine solely with our sequencing data. And our roadmap is by the middle of next year, so the second half of next year, to have 15 gigabases coming off the MiSeq. And these are 2 by 300 base pair runs. So those are 600 base pairs of actual sequence data, and then a variable insert size where you can have a virtual read length of exceeding 600 bases. And this is actually a conservative estimate. So this is what we're planning to commercialize, but we're already doing internal runs that exceed this. So it's not pie in the sky, it's real, and it's pretty amazing. And so this is an example of an internal run we've done where we've exceeded 20 gigabases. And that was with a 450 base pair run for the read 1 and a slightly shorter base run, 375 bases in the read 2. So know that commercializing this next year is not a stretch from where we're at right now. We just need to make sure that it's easy to run in customers' hands, that we've figured out any bottlenecks in the processing of samples. And then I just want to talk about sample prep methods and some of the new sample prep methods that might be of interest. So currently our mate pair library prep is really easy to do at about 2 kb, but if you get a library of any long, an insert in, now mate pair, just to fill you in, mate pair is what we call the long insert paired end library prep, and this is really useful for jumping over duplicated regions in the genome or helping cobble together these areas that are difficult to place in an assembly process. And so 2 kb is really easy to run, but the 5 kb libraries are a little harder to get a high diversity library. It just takes a lot of input DNA, and it's just more challenging to get a lot of data out of those higher libraries because you have to go through that gel isolation, and it's just not a fun prep. But we're launching a Nextera mate pair library prep, and we've got preliminary data on the mate pair library prep that will make it so much easier. So for those of you who are not familiar with Nextera, this is actually a transposon. Our adapters get added on by a transposon. So it's 90-minute prep, stem to stern, for our currently launched Nextera preps. We're also launching a PCR-free prep, and so that will be really useful where there are genome regions that are high in GC content, which right now, anytime you have a PCR step, you tend to get a little bit of bias away from GC. Our TrueSeq's pretty good, but there still can be some dropout of GC-rich genomes, so that'll be really important for plant genomes. Targeted resequencing aren't going to change. We have custom enrichment. And then the other one in red, which we're launching, is the TrueSeq stranded DNA prep kit for plant genomes. So this is specifically for plant, where you can actually trace it back to exactly what strand in your RNA sequencing data the expression tag came from, which I think is going to enable a lot of epigenetic understandings. And this just shows the PCR-free sample prep, and you can see that without it, there's some regions of the genome that are challenging to get coverage, and with the PCR-free, you end up eliminating that slight bias. And then last but not least, because we've talked a lot about at this conference about informatics solutions and the need for communities to work together to make sure that informatics resources are consistent and available to everyone so that progress can be made quickly, because we're trying to solve that 9 billion people question at NBQ. We've got, oh, sorry, skipping over this one. We've got base space, and we're working with the iPlant Collaborative, which has gotten several shout outs during this meeting because it's a pretty impressive offering. Essentially, we're working with iPlant to understand how we might be able to coordinate together to support the efforts that iPlant has already launched in the community. So they've got over 50 applications available. And for base space, I think we've got 6 in there now, and we'll be launching over 15 in January of 2013. So a lot of these are in their final stages of implementation. And then I just finally want to invite you to like our Ag Consortia Facebook page for those of you that are in countries that support Facebook. And you can see now that we've got, on this screenshot, there was 88 people that like us, but we have over 100 now because my mother joined Facebook. So that was very satisfying. And finally, I'd just like to thank our distributors for allowing me to come and having me and hosting me and doing such a wonderful job, both Supari and Kwe Wan, who you have all met in passing out the MySeq forms. So I encourage you to talk to them. And I believe that this says Kabkun Ka, which thank you very much for having me. And I'd like to open it up to questions because we'd love to talk to you about what's needed for the rice community. I've sort of thrown out there what we've done for other communities, but I think that the rice community is very special in that you've got that pristine genome, you've got so much data available, and you've got such really aggressive goals for the next couple of years in terms of sequencing and epigenetics. So thank you very much.
Any questions for Dr. Lawley?
Are these on? Can you hear me? So thanks, Cindy. Just in case you guys didn't realize, we gave away the answers to the first 2 questions, and if you didn't capture them, the first one was the current run specification for the MySeq. As Cindy mentioned, 2x250 base pair reads, 8.5 gigabase outputs, and in 2013 we'll be going up to 15 gigabases with 2x300 base pair reads. Okay, so please do fill out those forms to ensure your chance of winning a free run. And if you have any questions for Cindy, please post them.
Hi, Mike. Hopefully I didn't butcher the description of your publication. It sounds like it's on.
Okay. Yeah, I had a question actually about the SNP chips, because I mean, some people are still curious, like that pricing structure. So in rice, I mean, we have like 5 major subgroups within sativa, and then we have also glabrima and then many wild species. And so some people are thinking, or trying to balance, like, would it make more sense to have like a high density universal chip, or to have like multiple like medium density chips? So like the high density could be 100K or something, and then each of those subgroups, essentially, if you had like 10K or 20K within this subgroup, would it make sense to combine them all on a universal chip, have multiple chips, or have something like core content plus the add-on content?